of all, I'm just so glad to have this opportunity to discuss the work of the committee with you all and to solicit your feedback. This is a really nice opportunity to do that, and I really welcome your feedback and thoughts on the work that the committee has been doing over the past several years. So if you could go to the next slide, Jennifer. So the leadership as of last fall consists of myself, Allison Harrell. I'm currently at University of Arkansas for Medical Sciences, and Carol Yock from Health Canada, assisted by Reagan and Melissa very ably. Next slide, please. And on this slide, we just wanted to point out that we've got over 30 participating organizations, and we have a generous representation of all of the relevant sectors. So you can see industry, academia, and federal agencies having a really active role in a lot of our projects. So we're quite pleased with the representation that we have. Next slide, please. The overall mission of the Genomics Committee, as stated on this slide, is to advance scientific understanding and application of genomic methodologies for risk assessment, and more broadly to facilitate discussion and dissemination of information on the use of genomics as a tool to characterize mechanism of action and facilitate safety assessment of drugs and chemicals. So you'll see these themes come up as we talk about the various projects. Next slide, please. On this slide, we've binned all of our major projects into 3 buckets, and we'll go into more detail on each of these. So we have a focused effort on biomarker discovery and tools and methods to evaluate biomarkers. On the right-hand side, we have several projects that are revolving around technical evaluation of various genomics biomarkers and other things. And then on the bottom, we have had several projects which are related to applying genomic information to decision-making in drug and chemical safety assessment. Next slide, please. And you'll see that we have broad expertise in a variety of different areas. I'm not going to read all of these, but we have quite a lot of expertise in molecular systems biology, sort of more classical toxicologic and pathologic techniques, as well as informatics, and a lot of expertise in risk assessment and in pharmaceutical safety assessment. Next slide, please. Okay, so now we'll delve into the various projects, and we'll go through these relatively quickly. We'll give you some highlights, some of the information related to why we embarked on various projects and what we see as our major impact from completing the various projects. Next slide, please. So the first one that we're going to talk about is mouse models of the human population. If you go to the next slide, really the key issue that we identified was that there has been a lot of information indicating that genetic variability plays a key role in adverse drug and chemical responses, but there's really not been many models developed that can accurately predict the role of genetics in adverse responses. And we were particularly interested in idiosyncratic or very rare responses that have been very challenging to predict and to model in various systems. So our goal here was to explore the use of a genetically diverse panel of mice. And here we called it the mouse model of the human population, but what this really is just a panel of inbred mouse strains. And we wanted to see if this panel of strains could help us to better understand and predict drug-induced toxicity, and we focused on the liver in our particular project that the committee embarked on. So our approach here was to conduct a study, which was a collaborative study extending an existing project that was started at the Hamner and with collaborations from Pfizer, and to also develop a workshop where we could have more of a dialogue around using these technologies for safety assessment. Next slide, please. So the workshop that we held was in 2012, we had very good attendance, and we talked about various types of mouse models or mouse population-based models and how different laboratories have been using this type of model to investigate drug and chemical safety risks. Next slide, please. But for our particular study, we focused on, I think you might have to hit the arrow one more time, maybe to get the genomics box to pop up. There we go. So we focused on isoniazid as a model hepatotoxic compound. It causes idiosyncratic liver injury. We had a variety of endpoints. But really the punchline here is that we were able to model diversity in responses, particularly as it relates to isoniazid-induced hepatic microvesicular steatosis, where we had resistant strains and susceptible strains, and the difference was based on their genetic background. We were then able to integrate information relating to genomic sequence variation, transcriptomics, so mRNA expression levels that distinguished resistant versus susceptible strains, and metabolomic markers that could separate resistant and susceptible animals as well. And then we're able to integrate all of this data into a unified hypothesis regarding the potential mode of action that occurs in susceptible strains versus the resistant strains. So that's what you see on the right. And if you go to the next slide, we actually published these data in Toxicological Sciences, and we were very excited to be recognized by the Society of Toxicology Board of Publications as the best paper for 2016. So our impact in this study is that we provided some evidence for using this population-based technique to mimic human diversity relating to adverse drug responses. We were able to come up with mechanisms of toxicity susceptibility that we otherwise couldn't using conventional models. So if we go to the next slide, what I'll talk about next are a couple of projects that we have looking at microRNAs and toxicological applications. The first project was led by Carol Thompson and Philippe Pinet. So the major issue here was that there's been a lot of enthusiasm for investigating the use of microRNAs, which are small stretches of RNAs with transcriptional regulatory function. And there's been a lot of interest in using these microRNAs as biofluid-based biomarkers of organ toxicity because they're protected in biofluids, they don't degrade very much, their sequence is conserved across species, so there's good evidence for translatability across species, even into the clinical space. So there's a lot of enthusiasm here, but there's also a lot of challenges. And so we held a workshop in 2010 to really discuss, well, what are these challenges that the field really has, where our particular committee could provide some value. I'm not going to read through all of these, but there are quite a lot of issues that we identified here, one of which we started to work on. So if you go to the next slide, one of the key issues that we found is that there's really wasn't established best practices for measuring microRNAs in various biofluids. And so what we conducted was an interlaboratory study to look at where we may be introducing variability in measurement of microRNAs in these three biofluids. And so we wanted to better standardize approaches so that we can standardize measurement techniques across laboratories. And so what we did here was we had a four-phase study. We utilized a model for cardiac injury, and then measurements were made for cardiac microRNAs in serum, plasma, and urine across multiple sites. We had about 12 sites in which these microRNAs were actually measured. And in the fourth phase, we actually assessed variability of the preamp step, which is a necessary step for measuring microRNAs due to their low abundance in biofluids. If you go to the next slide, this actually outlines some of the data for the phase 2 and 3 microRNA best practices. This paper was recently accepted for publication in Toxicological Sciences. So the left-hand side just goes through some of the workflow, and the right-hand side just shows some of the key data, which indicates where some of the variability is actually explained through various methods and the site of measurement and things like that. So all of these data will be made publicly available, and we think will have good impact for informing measurement techniques throughout industry as well as academia. Next slide, please. All right, so as I mentioned, we were able to identify a standard protocol for measuring the absolute quantification of target microRNAs in biofluids. As I mentioned, we have a publication that was recently accepted. There's another paper that is upcoming related to measurement at the lower bound. So where's that lower limit of quantification? And so that's being written now and will hopefully be submitted soon. We've also presented these findings at various meetings, including a workshop on microRNAs that several members of this committee were involved in organizing at last year's Society of Toxicology meeting. Next slide, please. So our other effort on microRNAs is really related to a project that we call the Rodent microRNA Atlas, which is headed by Aaron Smith at Eli Lilly. Next, please. So the purpose of this project is that we wanted to inform microRNA biomarker discovery efforts. So there's been a lot of enthusiasm, as I mentioned, for microRNAs as biomarkers. But what we don't know is what we don't have is a catalog of microRNAs that are uniquely expressed within various tissues in the body. And why do we want to know this? Well, if we see a microRNA elevated in the blood, for example, we want to have some confidence that it came from cardiac tissue if we think it's a cardiac biomarker. But if it's something that is expressed, in a wide variety of tissues, it might not be a relevant biomarker or specific enough biomarker for what we're looking for. So that was the goal of this project, and the idea again is to inform biomarker discovery efforts and qualification efforts. Next slide, please. Okay, so again, what we want to do is we want to get towards this idea of liquid biopsies. Can we assay for microRNAs in the blood and inform underlying pathology? Next, please. So there's been several microRNAs that have been proposed as specific biomarkers of several different types of disease states and pathologies, but there's really quite a lot of microRNAs that are under development now. So having this atlas could really inform tissue specificity moving forward. Next slide, please. Okay, so what was done was 23 different tissues were dissected from male and female adult Sprague-Dawley rats. RNA sequencing was performed on these tissues, and then data analysis was performed at multiple sites in order to figure out which microRNAs are specific to various tissue types. And on the next slide, we'll show you some of those data. So what you can see here is that a wide variety of tissues actually have uniquely expressed microRNA, and so that's that is denoted by those green areas, shading on the different bars for each tissue. But also there are various microRNAs, and quite a lot of them that are tissue enriched, meaning that they occur in more than one tissue, but not many tissues. So there may be some potential to use a biomarker panel to rule out sort of off-target effects or non-specific organ damage. So this paper is, I think, under review at this point now. So a lot of this data has been put together. And if you go to the next slide, next, please. Great. So that paper's been accepted, and we started thinking about a second phase of work. So there's been quite a lot of scoping that's gone on within the past couple of months throughout the committee, where we've started to discuss where we want to go with this work. And some of the discussions that we have been around, exploring some of these specific microRNAs. Can we get a better handle on how specific they are, and can we go from the tissue level to even the cellular level to assess specificity. And then also there's been some discussion around exploring kinetics of microRNA release, because we know a lot of these microRNAs have a very short half-life, and is something like microRNA 122 for liver injury the best marker if its half-life is very short? Are there others with longer clearance half-lives that would be potentially more useful for the clinical setting? Next slide, please. Okay, so the last project that I'll talk about is one that is aimed at assessing adverse epigenetic effects of drugs and chemicals. We initially held a workshop on this idea, and we subsequently begun a study which is headed by Pfizer, and specifically Bob Chapin and Mike Lawton. Next slide, please. The workshop that we held was in 2013. It was very well attended. And the key issue that we wanted to sort of develop projects around is that several companies are now developing drugs that specifically target the epigenome as part of its therapeutic target. And what's kind of lagged behind are methods to assess safety. And in particular, we were concerned about epigenetic modifications that could be potentially inherited by the offspring. So we wanted to get a handle on what are these epigenetic modifications, and are there any modifications that are particularly adverse? Next slide, please. On the heels of the workshop, we developed a manuscript where we highlighted some of the key areas where we think that more data are needed. So what are the key knowledge gaps? What do we know now? And what data do we need going forward? And so that was published in the journal Toxicology. Next slide, please. Okay, so the project that we conducted was revolving around zebrafish. And in this case, we were looking at transgenerational inheritance of epigenetic modifications induced by drugs. So there's been some evidence in the literature that adverse effects of these exposures to these types of drugs and chemicals can be passed down to future generations. But a lot of these data were conducted in rodent models, and as we all know, rodents are, conducting studies in rodents can be time-consuming and very expensive. And so we identified zebrafish as a potential model which could shorten the time frame for these studies while still giving us important information about transgenerational effects. Next slide, please. So the overall goals of this working group was to conduct a transgenerational study in zebrafish using a tool compound known to modify the epigenome, assess zebrafish as a model to detect adverse effects of these potential transgenerational epigenetic effects, and to assess the sensitivity and reliability of methods for detecting epigenetic parameters for changes due to this drug. So next slide, please. So here's an overview of the approach that the committee has taken. At this point, I think we're in the S1 generation, so we still have a little ways to go, but we've begun collecting some preliminary data over time, which looks very promising for using this model going forward. Next slide, please. So our anticipated project outcomes for this ongoing project are assessment of zebrafish as a potential model for epigenetic effects, particularly ones that are transgenerational, and figuring out which endpoints we ought to measure in some of these zebrafish studies for assessing adversity. So our potential impact for this project would be informing development of a zebrafish model for safety assessment of drugs that are designed to target the epigenome. Next slide, please.
So I'm going to jump in with our next project, which is development of experimental approaches enabling transcriptomic analysis of FFPE tissue. There were two phases to this project, and Chris Mason was the leader of the first, and Susan Hester was the leader of the second phase of the project. So next slide, please. So a standard approach for collecting tissues is, for histopathology, is preservation in formalin followed by paraffin embedding, which is known as FFPE. And there's a number of archives of FFPE samples around the world. So there's in fact millions of archival tissues that could be available for molecular profiling. However, there's the problem, and that's that formalin treatment leads to adducts and cross-links, which makes it hard to recover RNA and amplify for molecular analysis. And the RNA also tends to be highly degraded. So this project addresses the issue of trying to enable molecular profiling of these untapped resources, which have degraded RNA. The project goals can be split into two phases, phase 1 being complete and phase 2 underway. In the first phase, we hypothesized that RNA-seq would be an optimal method for assessing gene expression changes in FFPE tissues. And in the second, we're aiming to improve RNA quality standards and RNA quality period. So phase 1 involved a collaborative study in which liver samples of mice exposed to the chemical furan were collected alongside controls. And there was profiling of these tissues, which had matched frozen tissues alongside liver samples preserved in formalin for different periods of time. And these were profiled by quantitative real-time PCR, microarrays, and different RNA-seq methodologies. We followed up with going to the NTP's FFPE archive, so I guess what we might call the real deal, to see if the technologies that we refined in the first part of the phase 1 project could work on decades-old samples. So in the next slide, you'll see some of the data, main findings from our study. So next slide, please. These are just some heat maps and some general quality assessment images from RNA-seq data on the bottom right. In the top, you'll see some heat maps that show the results of the bluish colored on the left-hand side of the screen were the control samples, and on the right in the more purpley reddy colors of maps are the furan-treated samples. And you can see all the platforms. We were able to refine methods for analysis of the FFPE tissues and distinguish control samples from treated samples. If you look at these heat maps carefully, you'll see that the ribodepletion method that we developed, that we refined for RNA-seq, was the most effective for the query of these formalin-treated tissues. And we also found that the longer tissues were in formalin, the poorer the quality of RNA, which is what we expected. But that we could see the expected gene expression changes in the FFPE tissues, even from the 3 weeks in formalin that we expected to see from a furan treatment. So we took the protocols that we refined in the first part and applied them to these NTP FFPE archival blocks, and you see those findings at the bottom right. So we had 8, 19, and 26-year-old archival samples, so decades-old samples. And these are quality output metrics, just showing what genomic elements we could identify in the RNA-seq data and the GC content. And we didn't identify any effect of time and block, suggesting that even in decades-old samples, we'd be able to produce high-quality RNA-seq data. The next slide shows the publication that resulted from this work, which was in Toxicological Sciences last year. And within the manuscript, we described best practices for the use of toxicogenomics approaches like RNA sequencing on FFPE tissues. And this really led to a proposal for phase 2 of the project. Next slide, please, which is described there. So one thing that we noted in the first study was that the standard methods used to assess RNA quality, such as the RNA integrity number, they weren't really useful in these samples because they're just so highly sheared. Bad was bad, basically, based on these RIN numbers. And we all know it's important to have a good quality metric. And so in this phase of the project, we really wanted to identify a good RNA quality assessment metric that we could use to identify which FFPE RNA samples would be of high enough quality to go on for a sequencing analysis. And in the second phase of the project, a variety of protocols and papers have been published that recommended methods to rescue or improve RNA quality from FFPE samples. And so we want to test these methods to see which of any of them are most effective. Next slide, please. So our approach in this project is described here. So this project, once again, leverages an existing experiment that was conducted at the US EPA by Susan Hester and Charles Wood. And within this project, mice were exposed to phenobarbital, a chemical with a very well-established mode of action. And the liver samples were collected and subject to various different preservation methods. So we have a frozen group, we have a section of liver preserved in ethanol, and then fixed in formalin for different periods of time. But this time we extended it up to 3 months, which is a realistic scenario in some of these archives. And then a subset of those samples that were treated in formalin were subject to 4 different types of demodification protocols. So the first part of the project, we wanted to develop a quality assessment protocol, and we evaluated several and settled on the DV200, which measures the percentage of RNA fragments over 200 nucleotides in length. And we found that this was effective in distinguishing subtle differences in RNA quality between samples. And so currently, all of these samples described in this image here are undergoing RNA sequencing analysis, and by September we're expecting to have the results of those come in, and so we'll have a lot of data to analyze for this project. And so we hope that this project will have a major impact in being able to access these previously untapped archival tissues. Next slide, please. And I'm now going to describe our qualification of a genomic biomarker approach for providing mechanistic context to positive findings in vitro chromosome damage assays. And this is the committee's longest running project and has been led by a number of different people shown here on the bottom right of this slide. Next slide, please. This slide speaks to the main issue that this particular project addresses, which is really the limitation of current approaches used to cancer risk assessment associated with genotoxicity testing. So on the left side, we have a description of how genotoxicity testing is done and the cost of the battery. And the limitations include that it doesn't address these in vitro tests for genotoxicity, although they're highly predictive of chemicals that may have a mutagenic mode of action in cancer, don't predict non-genotoxic mechanisms. But the biggest problem is really that there is a high sensitivity to these tests, but low specificity. And this leads to a number of what we call irrelevant positives. So these are particularly resulting from positive chromosome aberration findings in vitro that are not manifest in vivo. And this leads to risk management problems. It's costly and time-consuming to conduct a 2-year cancer bioassay, for example. And also, the assay doesn't provide mechanistic insight, which could be useful for human health risk assessment. The next slide, please. So to address the issue, these problems, the working group wanted to evaluate the transcriptomic biomarker to provide mechanistic context for in vitro positive genotoxicity findings. So to do this, this project leverages a biomarker that was developed in a joint project between Pfizer and Georgetown University. And that group developed a 65-gene transcriptional biomarker that they called TGX28.65. And this biomarker can be used to distinguish chemicals that induce DNA damage from those that do not. The goal of the project was thus to engage stakeholders in qualification of the transcriptomic biomarker approach for providing risk and relevance assessments for regulatory use. And we did this via the FDA's biomarker qualification process, so their program. So far the group has designed a study protocol and issued a letter of intent to the FDA, and then submitted a qualification plan to the FDA and had a face-to-face meeting with their qualification review team. And we adopted their feedback into the experimental design. The project has completed the experimental program of the work, conducted the data analysis, and we're nearly finished preparing the qualification package. And because the program has been running for so long, it's been presented at a variety of scientific meetings around the world. Next slide, please. So in the next few slides, I'll just describe a little bit about this, this biomarker qualification process. The top of the slide describes the experimental approach, which I won't go into. But based on the feedback from the FDA, we had 3 phases to the project, and all of these phases are completed. So in phase 1, we evaluated the technical reproducibility of the biomarker across time and across laboratories. In phase 2, I'll show you some data from phase 2. We evaluated the performance of the biomarker using 42 chemicals across diverse mechanistic classes. And in phase 3, we looked at the utility of the biomarker in cells co-treated with S9. So the biomarker was developed in human TK6 cells, which are not metabolically competent. So we wanted to look at integration with a metabolic activation system. And we also evaluated performance in a metabolically competent human cell line, the HEPA-RG cell line. The next slide shows some data from the project. So these are the results of the phase 2 part of the project. It shows some heat maps. On the left, that's the original biomarker with the genes on the y-axis and the chemicals on the x-axis, split into genotoxic and non-genotoxic chemicals. And you can see that the signature within the heat map is very distinct between the genotoxic and non-genotoxic chemicals. And on the right, these are the 42 compounds across the 5 mechanistic classes that are shown at the bottom, with class 1 being genotoxins that interact directly with DNA. So these are the established positives. Class 4 being clear negatives, and class 5 being those irrelevant positives. So I don't have time to review what we found in terms of the performance of the biomarker to distinguish DNA damage-inducing chemicals from non-DNA damaging, but I just wanted to highlight that the class 5 is really what we were gearing towards, and the biomarker did an excellent job at identifying relevant versus irrelevant gene tox findings. So here you can see these class 5s were primarily identified as non-genotoxic based on the overall score in blue just above the heat map. So the next slide, please. Once we had these data, we were able to propose a scheme for the incorporation of the transcriptomic biomarker into genotoxicity risk assessment of Ames negative compounds. So here the committee proposed that if you have an in vitro chromosome damage positive response, that this would be where you apply the transcriptomic biomarker to differentiate these relevant and irrelevant findings, and then you would follow up as appropriate for the risk assessment question you have at hand. The next slide just shows the timeline from the project from inception to present. As I mentioned, it's the longest running project within the committee. It was launched in 2007 with foundational work exploring time and dose response in the human TK6 cells. And then moving through our applications to the FDA, to the experimental conduct, and we're now looking forward to build upon this qualification effort. Next slide, please. Okay, so this is the current status. The project team has been meeting regularly to finalize the qualification package, and I'm really happy to say it's pretty much done. I think we'll probably have one more teleconference and finish a couple more things up. And we will probably be submitting this package at the end of the summer in 2016. We've presented all over the place with this project, and most recently at the July 2016 Toxicology Forum, and upcoming at the October 2016 International Congress of Toxicology in Mexico. And the committee, of course, will continue to interact with the FDA as the review package proceeds. So the next slide, please, describes beyond the qualification effort. I'm really happy to say that this qualification effort and the papers that have emerged associated with this qualification effort have received significant international attention. So for example, because this has been a HESI exercise and because of the FDA part of this project, Health Canada regulators have been interested in the biomarker, and we've conducted case studies at Health Canada exploring how we might use it in the assessment of environmental chemicals. And in fact, one of our papers applying the biomarker at Health Canada in collaboration with the committee was voted the best paper advancing the science of risk assessment by the SOT's Risk Assessment Specialty section. We've also collaborated to ensure integration of the gene set into the NIHS Tox21 S1500 list. We've worked with the NTP, the NIHS, to have a database and an analytical tool that we've developed for rapid assessment of the TGX28.5 signature in cells. Georgetown has been able to leverage the existing work to develop an SBIR grant to develop a high throughput method for genotoxicity assessment using TGX28.65. There's been interest by a number of different groups, including HESI's GTTC committee, so we're now in discussions with them to collaborate in the context of their mode of action working group in applying this biomarker for genotoxicity assessment. And we're looking forward to other applications, such as the committee has developed a grant for developing an antigenicity signature leveraging these findings from this project. Next slide, please. So this is the final project that I'll describe, and the committee has a working group that's focused on developing a workshop on genomics and cancer risk assessment. And the leadership of this committee is shown at the bottom. If we go to the next slide, this is the brochure shot of the workshop, which we hope to be held in the spring of 2017. So it will be a 2-day workshop. Next slide, please. Reviews the workshop goals. So to identify areas in the current cancer risk assessment paradigm where genomics approaches will have a chance to add value in the future. So we hope to have multi-sector stakeholder discussions to discuss applications and insight gained through application of genomics in this area, identify the roadblocks, and formulate recommendations. And of course, we'd like to publish this in a peer-reviewed publication, and we hope that it will enhance the use of genomics in weight of evidence assessments in cancer risk assessment. Next slide, please. And so finally, that brings us to our recommended disposition and looking forward to the next charter period. Next slide, please. So we are requesting a recharter for a 3-year term in order to complete our current activities. And I will review just very briefly what those current activities are to remind you. And we hope to be able to, over the next 3 years, also explore and develop additional project ideas. Next slide, please. So currently, we would like to continue to work, of course, on the genomic biomarker qualification package and obtain feedback from the FDA and move forward with the qualification process and submit a manuscript associated with this work as well. We will continue to complete the work in the microRNA biofluids project and channel these findings into the Phase 2 microRNA Atlas program. We want to coordinate efforts around the Phase 2 microRNA Atlas program, and we're doing that right now. We're in discussions. One of the areas of discussion is further characterization of candidate microRNA biomarkers identified in the Atlas to address biomarker gaps. We'd like to continue our experimental work in data analysis on the FFPE project and the Zebrafish epigenetics project. And in 2017, we hope to hold the Genomics and Cancer Risk Assessment Workshop. We're currently also considering embedding a number of new project ideas. Next slide, please. And looking ahead, as I mentioned, we are working on developing Phase 2 of the Atlas program. We will be spending time at our fall meeting in November discussing follow-up activities to the biomarker qualification effort. We're also exploring clinical translational elements of the FFPE NGS project, specifically with new members of our group from the National Cancer Institute. And most recently, we're interfacing with HESI's GTTC and CT Tracks committees on potential synergies on genotoxicity and assessment of genome editing therapeutics, which is a current gap in testing. So this could be a potential, very exciting collaborative project. And the next slide, please. Okay, so this is essentially our last slide, and this slide just shows the time frame of our current activities that will extend into the recharter period. So you can see we've been really busy, but we still have a lot to accomplish and to complete in the next 3 years. Overall, we think that the committee has provided valuable leadership and has had a positive impact in a variety of areas in genomics, in the area of, in particular, risk assessment. This includes furthering our understanding of the strengths and limitations of the use of genomic tools in risk assessment, enhancing our understanding of models to which genomic methodologies can be applied, and also developing new biomarkers and tools for risk assessment. Next slide, please. I believe that's the end of the presentation. So thank you very much for listening, and we're looking forward to